Okay, so we're going to finish off Tuesday of week 2 by talking about aerobic metabolism. Remember, anaerobic means without oxygen, aerobic means with oxygen. So we're going to talk about Krebs and electron transport. And again, most of this will be review, but we're going to go a little bit more in detail than we did in 220. So the Krebs cycle is just what it sounds like. It's a cycle. It can start wherever, but we generally think about it occurring with or starting with acetyl-CoA. Now, these processes occur in what we call the mitochondrial matrix, which is essentially the fluid, the gel, however you want to describe it, that's inside each of the mitochondria. Now, acetyl-CoA isn't formed at the end of glycolysis. In order for us to have acetyl-CoA, we have to have pyruvate, which means oxygen is present, and then that can be converted into acetyl-CoA. Okay, now remember with glycolysis, we form one molecule of glucose results in 2 pyruvates. Each of those pyruvates can then be turned into acetyl-CoA. Each of those acetyl-CoAs can go through the Krebs cycle. And each time it does, we form 1 ATP. Okay, and so you can see that here. Here's the ATP. All right, sometimes it's GTP, but the same type of thing, like we can break down GTP into GDP and get energy as well. The other thing that is important to know is that we form other molecules like NADH. So we've got 3 of those here, right? And then we also form FADH2, just like we formed NADH during glycolysis. And these molecules, both NADH and FADH2, will be used in the electron transport chain where we actually get a lot of ATP, get a lot of bang for our buck. And then the last thing is, notice that there's no oxygen required in here. You don't see oxygen going into any of these reactions, but we do produce a little bit of CO2. Okay, we don't produce CO2 during glycolysis, and we didn't use oxygen during glycolysis either. So Krebs doesn't need oxygen to occur, but without oxygen, we don't form pyruvate, which means we don't form acetyl-CoA. Now, with the Krebs cycle, it has a rate-limiting enzyme, just like the ATP, PCR, and and glycolysis. It's called isocitrate dehydrogenase. Okay, it means it's going to take a hydrogen away. And this is an enzyme that's the third step, which is ironically the same that PFK was. Now, the reason it's considered the rate-limiting enzyme is the same reason that PFK was considered the rate-limiting enzyme. Okay, it's sensitive to the concentrations of ATP, ADP, and phosphate. If I have high levels of ADP and or phosphate, then isocitrate dehydrogenase will be very active. If I have high levels of ATP, then that enzyme activity will be limited. And for the third time, this is our negative feedback, or this is an example of a negative feedback loop. And so this leads us into the electron transport chain. And the electron transport chain is complicated. I think it's more complicated than the other processes that we've talked about before. Now, this occurs along the inner membrane of the mitochondria. Okay, so what happens is, and I'm going to kind of give you a brief overview, and then we're going to go a little bit more in detail, is that the NADH and FADH2 carry electrons to the electron transport chain. And then these things undergo a series of reactions to ultimately produce a lot of ATP. So to start off, each reaction involves the movement of electrons. As electrons move down the chain, energy is released. Okay, so they go from, where'd my cursor go? Okay, so they go from here to here to here, and so on and so forth. Okay, ultimately, these electrons need a place to go, and that's oxygen. If there is no oxygen, there's no place for the electrons to go. They don't just like hanging out by themselves, and so there's no ATP formation. So this is why oxygen is so important. It will hold on to a pair of electrons. One molecule of oxygen can hold on to a pair of electrons. And ultimately, what happens is, and you see it right here, is that we form water and we form ATP. Okay? So, kind of the same way to look at this is that we're delivering electrons by NADH and FADH2. And NADH and FADH2 come from glycolysis and Krebs. And so what they do is they transfer electrons at the beginning. And so this actually causes them to turn into NADH and FAD. Okay, and so we see in this picture here, we've got NADH that's now NAD. Okay, as electrons are passed down the chain, they move from a high level of energy to a low level of energy. And so some of this energy that's released pumps hydrogen ions, moving them out of the matrix, so the mitochondrial matrix, which is here, and up into this inner membrane space. And now we've created a gradient. Okay. Now at the end of the electron transport chain, electrons are transferred to oxygen, which we talked about on the last slide. And so that's happening right here. And we form water with that. Okay. And as these hydrogen ions flow down their gradient and back into the matrix, so that's right here. So they're going back down because high to low. They pass through this enzyme called ATP synthase. And so what that does is that causes ATP to be formed. Okay, so again, I know it's complicated, but ultimately what's happening is that we're moving electrons, and by moving these electrons, we're creating a gradient such that hydrogen can come through this last protein and allow for the formation of ATP. But these electrons will not move unless they've got somewhere to go, and that somewhere is oxygen. Okay, so why do we care? Why is this important? Well, one molecule of NADH produces about 3 molecules of ATP. One molecule of FADH2 produces 2 molecules of ATP. Okay. Now, when you read your textbook, I believe this textbook talks about how one molecule of NADH produces 2.5 molecules of ATP, and FADH2 is 1.5 molecules. Okay. There is some ATP that has to be used here, and so there's some adjustment there. That's where that 1.5 and 2.5 come from. Okay. I generally just like keeping it nice and easy, 2 and 3. It's just a lot easier to remember. But that's why you might see a difference. Now, just like there isn't necessarily a specific enzyme that we inhibit, but if I've got lots of ATP floating around, the electron transport chain doesn't work as quickly. Okay. But if I've got high ADP or high phosphate, that's going to activate the electron transport chain. Okay. And so if you go back to the start with glucose, so one molecule of glucose, we're going to say forms 2 molecules of ATP in glycolysis. So I'm just going to put a G here. 2 more molecules with Krebs. And then we form 34 molecules in the ETC. Okay, and that's because we've got 10 NADH and 2 FADH2. So 10 times 3 is 30. 2 times 2 is 4, so 30 plus 4. So 1 molecule of glucose gets us about 38 molecules of ATP. All right.